All right, we have the next panel coming up, and I am going to invite in Stephen Uzzo, a long-term collaborator. So glad he could take on that panel and organize it and moderate it. And there are a number of panelists. So I'm going to promote Scott Echols and also Olson. Amazing that you could join. We have Joe Heimlich, also a long-term collaborator from the museum world, Liesel Record, which is a dear friend and colleague, and Yoon Zhang. And we actually met in person, Yoon. I don't know if you remember, but I was fascinated by your work, and I'm so glad to see you again here. So, Steve, I think that's all of them. Do you miss anyone? I know it's the biggest panel we have in the entire 24-hour session. I think you got everybody, right? So I'm going to disappear, but have fun, and I'm going to watch you. Cool to see you all.
Okay. Weidi might not be on yet, but hopefully she'll join any minute. So thanks, everybody, for joining. I presume everybody knows about this event and the fact it's going for 24 hours, and it's about the Human Reference Atlas project that aims to map all the human cells. There's Weidi. Thank you for joining, Weidi. And all the cells of the human body to advance biomedical research and clinical practice. For this part of the tour, we'll be exploring ways we might bring the Human Reference Atlas to the public. We have 6 experts from diverse fields to participate with all of you in discussing their work. The goal is to acquaint you with some of the compelling ways that complex ideas a human reference atlas represents can be experienced across sensory and intellectual domains. And there are a variety of ways that informal science learning through exhibits, visualization, modeling, and other innovative modalities can inform the public of the value of this work and provide new perspectives and tools to advance biomedical, educational, artistic, professional fields of endeavor. So I'm Steve Uzzo, recently of the National Museum of Mathematics in New York City, and previously the head of science for the New York Hall of Science, also in New York City, and I'll be the moderator. So I guess we can start and introducing. So Yoon Chung Han is assistant professor at the Department of Design at San Jose State University. She is an interaction user experience designer and multimedia artist, as well as a visualization researcher. And Yoon, I think one of your movies might not be running properly on the slide, so we'll give it a try, and but we can also give you the helm and have you present from your computer if that's easier. So this is Yoon Chung Han's presentation. Do you want to start?
Sure. Yeah. Thank you, everyone. Thank you for having me. So today I'm going to talk about my work focusing on the biometric data visualization and sonification. So I've been working on the biometric data visualization and sonification in the past, I think it's been like a while. I think almost 10 years, actually. Can't believe it. So yeah, among many actually tries and approaches, I'm going to actually show 3 works. I made it, and then could you go to the next slide? So if not, I can actually play the video from my computer. Okay, yeah, I'll share my screen.
Okay.
Yeah. All right. Okay. All right. So, okay, do you see my screen?
Yes.
Yeah. All right. So the first work is it's called the Dissonance. So basically, it's an interactive fingerprint sonification and then sound installation that transformed humans' fingerprints into musical sound. So the idea is to allow audience to explore their own identities through unique sound generated by their fingerprint patterns based on algorithm computing and the physical device. So as you can see in the video, you can actually choose any kind of finger and then input, and then you'll see the 3D animated visuals on the screen. And then at the same time, you can actually listen to the sound. Let me make it a little bit. Do you hear sound? Or a little bit?
Yeah. Sorry, not hearing sound.
Okay, actually not then.
Can you play it on your computer so we can hear it through your microphone?
Yeah. Okay, let me try one more time. How about now? Your sound?
Yes.
Okay. I know there's some kind of little bit noisy sound. So basically, the fingerprint will become an input data, and then you can actually listen to all kinds of sounds, because actually everyone, we have all the totally different fingerprint design, right? So I use some kind of minutiae and then some kind of bifurcations of the lines, and then many visual features. You can actually get it from the fingerprint, such as the size and the angle and then brightness level. Those kind of all the visual features actually are mapped to the sonification, and then you can listen to it. I know we have actually 5 minutes per person, so I'm going to actually move a little faster. So this is the second work. It's called Eyes. So it's also interactive art installation. So the interaction-wise is pretty much similar. So you can scan your iris through the camera station, and then unlike the fingerprint is mostly like focusing on more like a line analysis and then patterns. But this way, we have beautiful colors in our eyes, iris. So my customized software actually kind of analyzes all the kind of color data, color info from your iris, and then make a dominant colors. And then colors are actually used as a kind of core, like a frequency and pitch of the sound, and then generate the sound. So you can a little bit listen to the sound here. So here, there's actually interactive component, and the next to the installation, there are actually 3D printed sculptures. So the audience can actually listen to the sound, compare with their iris sound, and then it actually replies the sound generated from the iris data, and the project 3D converted images on top of it. So the audience can listen to it and question the problem of in disembodied identities in the digital art, and then digital era through the existence of the audio visual representations of the individuals. So luckily, actually, I had a chance to show this work in many places. One of them was actually ITRVs, where I met Kathy before. So yeah, when I exhibited this work, I always actually made a lot of beautiful eyes that had really obviously represent diversity or our, all human beings. So I think I always mesmerized by looking at the eyes and different colors, and then when I also made the really cool colors, I feel really thrilled. And then it's also amazing to listen to the sound. So this is actually in the SIGGRAPH gallery in 2019. And then actually some people actually took some photos that are actually kind of distinctive colors. So third one, the last project is called Rose and You. So this is actually also interactive biometric data artwork, actually focusing on the veins. So it actually allows participants to scan their veins and then find the rows that match to their vein lines. So the vein data is one of the fascinating forms of the biometric data that actually contains the uniquely complicated lines that resemble the roads and paths surrounding us. So I think rows and the veins have some similarities and also differences. So that kind of correlation between two different data was actually the point in this project. So this new artwork actually explores the line segmentation and structure of the veins, and then compares them with the rows in the real world. The participant can also export the data and they keep them as a personalized souvenir as a 3D printed sculptures like this. And then actually I recently actually showed this work in the current new media festival, and then some people actually asked me to print it, so I actually printed and then they actually purchased it. So it was a pretty cool experience. So I think this project has the potential to lead the way in the interpretation of complicated data sets while providing aesthetically beautiful and mesmerizing visualizations.
Wow.
This concludes my presentation.
Wow, Yoon, that's amazing. I know Kari posed the question, where can viewers experience these installations? So maybe provide some links toward the end of the presentations after, and we have question and answer. People may have other questions. But I just want to keep us moving along. So the next speaker is Joe Heimlich. He's Senior Director of Research in the Center of Science and Industry and Research for the Center of Research and Evaluation at COSI. Joe is also a Professor Emeritus of Ohio State University, and as an EdPsych person. He focuses his career on how people learn in zoos, nature centers, parks, museums, science centers, and other informal contexts. He's worked in the environmental learning field for over 40 years as an educator and researcher. Joe, I'll bring up the presentation.
Well, that's coming up. I'm going to take this a slightly different direction because my work is less in the generating and more in the understanding of how people take in information, take in the data, and make meaning or don't make meaning of what we're trying to share. So what I wanted to just say is the visitor in human body exhibits is really my interest. And why the human body in exhibits? There have been a lot over time, hundreds and thousands of examples of using the human body as whether it's art or as a science tool or any of those other ways that we might view human bodies. Some of them have included things such as the body image and alterations of the body and shifts to body image, body as a vessel, body as art, and a lot of that in the 20th century. Body as a lesson, the historical way of presenting the body in like science museums. The body is history, looking at the change of people over time and the change of a person over time. And finally, the body is a tool. How does the body function and what does the body do? And so we've seen a lot of reasons why there have been exhibits or how the body has been exhibited. But I'd like to raise some questions. Next slide. So in learning from exhibits, there knowledge, attitude, and behavioral learning across the spectrum, there are really two structures of learning that apply in these kinds of contexts. We're looking at the non-formal and the informal learning, the learning that is controlled informationally by an institution or organization, but that the individual chooses the ways in which or chooses to or not to engage in the learning itself. Theoretical bases for learning in this area are those of much more aligned with adult learning. If anyone looks at how people learn and look at lifespan learning, much more how adults learn than how children learn is how people learn naturally in exhibits and in experiences. I find it very helpful to think of this as asking the 5 whys and the H. And in this case, it's really the 3 whys. We don't need to ask so much as the when or where. That's given in an exhibition or an exhibit. But it's the who. Who is it for? And who is giving it? What? What is it about? What is it we want people to walk away with? What is it we want people to hold as an understanding? And how do we want that to lead to a behavioral change in the person and in society? Well, why? Why do we do this? And maybe that is the most important question we can ask. Why do we feel it is important for others to get this information, to hold this information, or to have this experience and hold this experience? We often create with a sense of people will understand what I'm after simply by their experience of it. But when we're trying to share knowledge or share information versus share art, there's a very big difference in the why and in the what. And it may seem trivial to raise this, but it becomes a major issue and a major barrier for visitors to exhibitions or to experiences related to elements such as cohesion. How cohesive is this experience, and how does it tie to my life, to my prior experiences, and to where I'm heading next? The narrative itself, how do I understand this? How does it fit in the structure of my life? And how does this narrative lead me forward? How does this ground me? Understanding that I need to be grounded in both the physical and the emotional experience as a visitor, as a guest to an experience, and understanding that how we create an experience grounds the person or doesn't ground them to lead them to the outcomes we're after. And finally, how do we design this in such a way that it becomes a foundation for learning, and that it becomes an opportunity for people to actually walk away with understanding, with insights, and with things that they can hold and move on that are relevant to their lives. So that as we move into the dialogue, that is the position I'm going to posit myself into. Thanks, Stephen.
Thanks, Joe. That was really an excellent sort of circumnavigation of what it means to have to build exhibits and figure out what they're for and determine who the audience is and all of that. So I really appreciate that perspective. So we have next is Elizabeth Record, who is Associate Director for the Cyber Infrastructure for Network Science Center at Indiana University in Bloomington, where Kati is from. Liesl does strategic planning for the center as well as a variety of educational programs and co-curates the Places and Spaces Mapping Science Data Visualization exhibit. So Liesl, if you want to talk to us through these.
Great, thank you. I wanted to share a couple of examples from this traveling exhibit on data visualization, which I co-curate with Dr. Kati Borner and Todd Theriault, both here at Indiana University. The exhibit's called Places and Spaces Mapping Science, and it showcases leading examples of how to visualize information, which is why I think it's relevant for this conversation today. In particular, it focuses on ways to visualize the knowledge landscape of science. It's a 20-year project, and for the last 8 years, we focused on interactive visualizations, which we call macroscopes. These interactive visualizations are tools for presenting and exploring large data sets, which can be pretty challenging for the layperson to encounter and make sense of. So I just wanted to start by sharing 2 brief examples of some of the many visualizations in the exhibit. So if you could go to the next slide. This first one I wanted to share with you guys is called Virus Explorer. It was designed and developed for the Howard Hughes Medical Institute in 2016. And the purpose of this interactive is to provide students and educators with an opportunity to compare and contrast different kinds of viruses. What do they infect? Where are they found? What do they look like? It's part of a series of classroom resources associated with the film Spillover, Zika, Ebola, and Beyond, which aired on PBS. The data-driven 3D models that you can see here, you can expand those and interact with them. They allow students to make comparative observations. And in the process, they learn about the criteria that scientists use to classify viruses, what characteristics of different viruses are important, and the global prevalence of viral infections. So cross-cutting concepts in scale, proportion, and quantity are all designed to tie in with biology curricula for high school and college students. This award-winning visualization was developed in cooperation with teachers, which I think is an important point, and a place it's designed to tie into the curriculum. It's accompanied by a rich packet of additional resources that go with this to be used in the classroom. It won the Biocommunications Association's Best of Show Medical Education Award of Excellence in 2017. And it was also a finalist in the Vizi's Visualization Challenge that same year. So that's one example, kind of designed for the high school level. And then if you can go to the next slide, I'm going to show you another one. And this one is really designed for community planners and activists. This is called the Appalachian Overdose Mapping Tool. And it integrates overdose mortality rates for each Appalachian county with community factors like unemployment, poverty, disability rates. Users can compare county level information to the rest of the region. And you can see a visual representation of how that data changes over time. I think this is a 4-year period of data that's included in this macroscope. The tool was initially developed by NORC at the University of Chicago in collaboration with the Appalachian Regional Commission, an economic development agency of the federal government, and also in cooperation with 13 state governments in the Appalachian region. They've since released a national version that's called the Opioid Misuse Community Assessment Tool. So if this is of interest, you might want to look at the national one as well. The tool, as I said, was designed for community leaders to be able to look at correlations between different types of data. So it's a huge data set that's presented in a really interactive but easy to access way. For example, you can look at different data overlays to show correlations between overdose mortality rates and other socioeconomic and demographic factors like unemployment, poverty, disability, age. There's a whole variety of different factors you can explore. And in doing so, you can see, for example, in Central Appalachia, counties with the highest rates of overdose are often the same counties that have the highest rates of people on disability. Likewise, you also find that the counties with the highest overdose rates are often the same counties with the lowest rates of educational attainment. So as you start to see patterns as a community member or community leader, this may be start your wheels turning about how to address some of these issues. In Northern and Southern Appalachia, the highest overdose rates are in urban counties. So you can kind of test your hypotheses and see if what you think is happening, does the data bear that out? The simplicity of the interface is really pretty incredible given the complexity of the data set that's driving this visualization. It incorporates three main data sources. The CDC National Center for Health Statistics, National Vital Statistics System multiple cause of death file. That's even a hard one to say. And the second one is the US Census Bureau's American Community Survey. And the third is the Bureau of Labor Statistics quarterly census of employment and wages. So you can imagine there's quite a robust amount of data behind this one. That's just a brief taste because I know we have a lot of ground to cover today, but I wanted to show you just a couple of visualizations from the exhibit. If you like what you see here, feel free to visit the website at scimaps.org. And I also wanted to let you know that we're accepting macroscopes for the next iteration of the exhibit. So we have a call for submissions out. So if you have a health-related macroscope that you're aware of, we would love to hear more about it. Feel free to go to the website and submit your work. Thank you.
Thanks, Liesl. So our next presenter is Scott Echols, a veterinarian educator and founder of Scarlet Imaging, which develops contrast agents and techniques to greatly enhance anatomical imaging. He has particular interest in avian medicine and the creation of educational media to improve education about and health of birds. So, okay, so you're sharing. Is that working, Scott?
Yes.
Okay, good. Thank you. Carry it on.
Can you hear me okay?
Yes.
Okay, great. First of all, thank you so much for inviting me to be part of this. This is really exciting, and I'm listening to the diversity of these presentations, and it's really fascinating. So I'm very, very happy to be a part of this. So I'm going to go through this very quickly. What I'm really trying to do is find better ways to the human body and others. The first things I developed was a contrast agent that allows us to see down at the capillary level. So it's 100% non-toxic. We can inject it as a full body. The largest animals we've done so far, a full horse. We've done cattle, all the way down to zebrafish, and of course, lots of people. And this is compatible with all forms of imaging that we've tried so far. CT, all your X-ray technology, CT, MRI, ultrasound, light sheet microscopy, histology, videography, and all that kind of stuff. So we really these amazing tasks and
Scott, you're breaking up a little.
Tissues, and we can do a whole organism, or we can visual. What we depends on our ecology. Is that any better? Yeah. Am I coming in?
Okay. I was just breaking up for a little bit before. Scott, you there?
Is that okay? Okay. Yeah, I actually just landed, and I'm just actually yes. Can you?
Do you want to mute your video? Maybe.
Am I coming through? Uh-oh. Okay, let's try that and see if that.
Yeah, try turning off your screen.
Okay, so in this example, we can see down the capillary. Turning off the screen. Okay, let's see. Okay, see if that better, any better? Is this coming through at all?
Oop. Looks like we lost him. Let's give him a second to try calling back in. Okay, Scott, you're back on. We're not hearing your audio. Scott, you're muted.
Is it coming through at all?
Yes, you were muted. Thanks.
Okay. Sorry. We're looking at an image of a kidney and the inside of the vasculature of that kidney. And this is because we can look at the vasculature using bright view, micro CT scan, and get to an incredible depth because we're getting down to the capillary level. Now, we look at macroscopic structures such as this human vasculature level. We have done this every part of the human body so far, and we are looking at, this is why I'm actually on this trip, is to do more of this type of imaging so we can get a better accounting of different parts of human anatomy. And this has been fascinating to me as a veterinarian, seeing the tremendous variation in human anatomy. So again, we can combine to perform multiple imaging on the same subject, and it's richer patterns and so forth to look at the anatomy in greater depth. Now, the second part, and I'll go through quickly, is I've developed a way to preserve cadavers without the use of formaldehyde, such that the tissue lifelike, and we simply just true tissue. In fact, we can pump real blood through these donors, and we can get blood oxygen levels if we inflate the lungs and breathe for them. The kidneys will still produce death. And then again, we can do our imaging modalities. So this is an example of a human cadaver. This is only 67 days post-preservation, but you can kind of see how lifelike the tissues are. This gentleman had COPD, so his lungs are in pretty bad shape, but that's understandable. And then you can see the flexibility of the tissues that we can obtain. Now, not only are we doing anatomy classes with this, and of course, if we put bright view in there, we can scan and get those images. We're also using this for surgical training. Because the tissues mimic the real tissue, we can actually perform a whole variety of surgeries. So far, we've done everything from involved trauma surgeries to neurosurgery, brain surgery, a variety of soft and orthopedic procedures. So in this example, this is an adrenalectomy. And I'm going to zoom ahead here and using the Da Vinci robotic system. And this is the adrenal gland right here and cutting it away from the vena cava. So it does allow for a whole different level of surgical training. So I know that my video is unstable, so I'm going to kind of go to the end here and we'll let you continue on. So thank you.
Wow, Scott, that's amazing. And we really appreciate you taking time to, I know it was a big rush to get to the hotel and we had trouble with the audio and everything, but really great to see your work and looking forward to seeing more. It's fantastic. So let's move on to our next speaker, who is Arthur Olson. He's the Anderson Research Chair and Professor, Department of Integrative Structural and Computational Biology at Scripps Institute, and is founder and director of its Molecular Graphics Laboratory, which focuses on the development of novel and intuitive human interfaces for research and education in structural and molecular biology, utilizing solid printing and augmented reality technologies.
Okay, yeah, thanks, Steve. And also Katie and the other organizers. It's been a very interesting series of talks through the day. I haven't seen all of them, but the variety and the scale and the expertise is quite remarkable. So I come at it from a very different point of view, which is from the bottom up, from atoms to cells. My lab comes out of a structural biology background, but has a lot of kind of visualization and even some art in it. The point that I'm working from is that we now acknowledge that we live in what we call a bioatomic age, where life, we can start to understand life from the molecular viewpoint. We have the concept that there's a physical continuity from the atom to the organism. And the modeling of molecular mechanisms of life becomes feasible. And we have to understand that it's all physics, but made more complex by the combinatorics of chemistry, and made even more complex, much more complex, by evolution, which actually embeds history of the molecules over time. So, and we have different types of models for each of those domains, but biology really requires multi-scale, multimodal models. I like the analogy of the simplest cells that we know, bacterial cells, have on the order of 2 million proteins, which is about the population of the island of Manhattan. And in both cases, we'd like to know what's there, where is it, how does it work, and why is it doing what it's doing?
Utilizing solid printed models
so
of virus
this is an
components
example.
And magnets representing the attractive forces between them, we can produce a physical analog capable of self-assembly by random shaking. The model demonstrates not only how nature accomplishes this feat, but can show some of the subtle characteristics of stochastic assembly. Notice that the subassemblies form and break apart en route to the most stable structure. The strength of the shaking is analogous to the temperature of the molecular environment. If it's too hot, the virus will break apart. If it is in the proper range, assembly will take place at an optimal pace.
So this is an example of how you can demonstrate something, a relatively surprising concept, which is order from chaos. And how does life put itself together? Now, this type of physical representation has a lot of power because you actually see it. I put this movie onto YouTube, and some of the first comments I got was, "Oh, you just shook it apart and ran the movie backwards." You can't do that if you do it yourself. So there's a lot of power in basically physical models. Now, why are we interested in life at this level? Well, an example I would give is my colleague at Scripps determined the structures of the spike protein of SARS virus and saw the difference between its pre-fusion state, when it's floating around in the blood or wherever, and the post-fusion state, where it exposes the receptor binding domain and can enter the cell. Now, the immune system is most effective when the virus is free in the pre-fusion state, but it's not. It basically is a shape changer. So, with the knowledge of the structure, one can find out what that mechanism is, which is kind of a jackknife mechanism that changes the shape. And from the sequence and knowing the structure, you can pinpoint where the hinge of that jackknife is. And by changing 2 amino acids, you can lock it into the pre-fusion state. And that is the engineered antigen of Pfizer, Moderna, and in fact, almost all of the vaccines that you hopefully have in your body. So these vaccines are based on that kind of knowledge, and they have that kind of impact. Another example, just to highlight kind of how you can put these models together, because they are models. It's a synthesis of structural information, proteomics, genomics, metabolomics, etc., into a three-dimensional model of a complex biological unit. This happens to be the insulin secretory granule in the pancreatic beta cell. You can see the insulin crystal in a maturing vesicle, but there's different states of that vesicle. What you see on the right is the proteome of this particular structure, both what's inside and what's on the membrane. So these models, we have developed tools to build these kinds of models at the real density and population and shapes of the molecules that compose it. And using that, so how do we use information like that? Well, basically, what we can do is take a low resolution. The data for the cell is relatively low resolution. You don't see atoms in cellular structure using current methodology if you're looking at intact cells. On the other hand, soft X-ray tomography, for one, can give you about a 40 nanometer resolution. So you can see these vesicles and you can measure kind of vaguely the intensities, but you can't determine from that raw data alone the state of those vesicles, whether they're immature and don't have any insulin crystal in it, or whether they're mature and they're ready to leave the cell and distribute the insulin. So by making models of all the possible states, using the software that can actually put these things together, we can simulate the soft X-ray experiment and put information into those experimental data to, in this case, color code them, whether they're mature or immature or what stage of maturation they are, in order to understand the process. And the last thing I want to mention is the kind of tools that we have that are interactive. Just like the shaking virus, I think that interaction and process is an important way to get information into a group of people. So many of you may be familiar with David Goodsell's watercolors. He's a colleague in the lab who's both a great artist and a wonderful and productive scientist. And he has changed the view of how people think about the crowdedness of cells. And each one of those paintings that he does, most of the work is not in the watercolor itself, but it's in the integration of the science that makes these models.
So Bart, can we move it along a little? Thank you.
Okay, yeah. So I just want to show that we've developed a tool so that you can do David type paintings, and this is sped up by about a factor of maybe 5. But basically, like David's paintings, all of the elements are of the proper scale. They go into the proper diameters, and you can develop complex illustrations, which is a learning tool in and of itself. So this type of tool is currently available online that people can use. It's just called Cell Paint. And currently what we're doing is we've taken the ideas of Cell Paint and put it into virtual reality environment. So you can basically construct structures larger structures from known smaller structures. This happens to be a spectrin network on the surface of red blood cell. And you can actually be in there. You can be drawing new structures. You can be connecting them and so forth. And with that, I think I'll end. Sorry for going over my 5 minutes, but.
Great. Thank you, Art. I know it's a lot to talk about in a short period of time, and all of your work looks incredibly promising. We're just going to move along to Weidi Zhang, who's a new media artist and designer and assistant professor of immersive experience design at Arizona State University. Her interdisciplinary art and design research investigates a speculative assemblage at the intersection of immersive media design, experimental data visualization, and interactive AI art. Can you release the screen?
I'm trying. Let's see. I'm looking. I see. I'm in the, okay. Sorry. It just says resume share. Somehow I'm stuck. Can you unstick me? Am I still sharing?
Yeah.
You can't stop share. Sorry. Okay.
Go.
Sorry about that.
Okay, Weidi, thank you.
Okay.
You have a presentation. You're going to share your screen now, right? Okay, thank you.
Am I sharing my screen?
Yes, I can see it.
Okay. Does the sound okay? Did you hear me okay?
I can't hear a sound, but I can hear you.
Can you hear my voice?
I can hear your voice.
Cool. Thank you. Okay. Hello, everyone. My name is Weidi Zhang. I'm from a new media art and design background. And today is my great pleasure to join this virtual panel and to share my artworks on the topic of interactive artistic visualization that is co-created by humans and machines. So today we are living with machines every day of our life, and the machine can provide a new level of automation for the design of artistic visualization for the human bodies. And countless surveillance cameras are hidden in the public space where our life moments are transformed and our body are transformed into data, which is constantly evaluated by intelligent machines. So my research proposes an interactive AI system for visualization that includes an imaging device in the art installation to capture the participants in real time and send the live streaming to the customized AI application, which interprets the live streaming based on neural networks' prior knowledge and output information, which is further processed with culture and aesthetic decisions to generate new visualizations, which will be presented in the gallery space as the art experience can be perceived by participants. I want to emphasize on the three contributions to the interactive visualization: audiences, artists, and machines. The visualization can be presented in multiple formats and forms because images are programmable database. So it also extends to the immersive medium. For example, my interactive visualization are presented as VR art experience or as audiovisual Fudong performances in the planetariums. During the pandemic, people are keeping social distancing to avoid spreading the virus. So I visualized and materialized the voids between human bodies during the pandemic as data-driven sculptures. Inspired by the idea of yin and yang from Taoism, the positive and negative are producing each other. Taoists believe that everything is part of the oneness of the universe, and there is no difference between the internal process of our human bodies and the external forces of nature. So I rethink the relationship between the space between humans and the disconnection and the social transformation. So I create the second part of this project using the techniques of volumetric capturing to capture two participants in the art installation in real time and visualize the voice as an ever-changing visualization. The last work I will share today is called Ray. It is one of my recent interactive AI art experience, aims to address the changing ontology of the images under the gazes of machines. It creates an intelligent system that incorporates automation with originality to generate a visualization of human bodies. So it got inspiration from the rheograph. It is also called photogram. It is an image created by exposing light-sensitive photographic paper to lights, where the objects are placed on top of it to block the areas of the paper. The variation of this artistic technique are also implemented for scientific purposes, such as X-rays, which are made by using external radiation to produce images of our body and other internal structures for diagnostic purposes. So this art project makes metaphors for the power of gaze by linking the humans under surveillance cameras with the objects for photograms. And a neural network is trained to translate human portraits into new images of rheograph with semantic meanings through learning over 3,000 pairs of rheograms from 20th century and human portraits using Flickr API. And it also incorporates a computer vision system to output the semantic information of the visualization. In the art installation, the intelligent system Ray constantly observes participants via camera and transform the live streaming data to a novel semantic rheograph that evolves in real time. The father of information design, Will Bertin, who declared in an essay included in Swiss Design magazine that he pointed out man as a measure and a measurer. Human was the most important element in the design process because effective visual communication depended on the audience's physical, emotional, and intellectual response and comprehension. So my artworks trying to address the question of how can we create a critical future ideation through artistic visualization that is co-created by humans and machines. What kinds of poetic solution we can find from the art history for the visual representations of human bodies. And I was trying to create a liquid network to build a culture that blends human and computer meanings. Thank you so much. And here's my website, my art's Instagram, and my email address.
Thank you so much, Weidi. Just amazing stuff and very evocative. So this is an amazing group of people, and I know we're coming down to a very small amount of time to have an open discussion. But I guess we talked a little bit about some of the possible questions. So all of this amazing work that's going on that everyone's doing, thinking about how a human reference atlas, which is now kind of it's early on in its inception, what does it need to go forward? How could they apply some of these technologies to bringing some of those data that are going to be so enormous to life for people? And I know you've demonstrated some of the ideas, but do you have any further sort of conjecture is like, okay, if we took the way I'm thinking about this to this human reference atlas, are there sort of ideas that it might evoke in you? And anybody can answer that question, please.
Well, one thing that I'm taken with is the interpretation of the human bodies, the sonification and the photogrammetry engages people. So it seems like if you want to get people to be aware of this, the work of the Cell Atlas in a kind of a museum type or broadcast type setting, the more you integrate their own bodies into it, the more interest you'll get. So that's one takeaway that I have from the discussions today in this panel is that who's your audience and what do you want them to take away?
That's great, Art. Thanks. And Scott, you had a comment.
Yes. So there's a couple. This is a huge topic, really, and we have to and I've seen some beautiful representations, like Art was saying, the artistic side. The human body is, amazingly beautiful structure. It's also highly variable. And when we look at our current atlases, if we just take what we have right now, it's an artistic representation of one body type, if you will, and that's put in paper. And I look at and think about, how do we take all this data and really analyze it to maybe even what does the kidney look like from a 50 to 60-year-old woman of this malady? And really, I think data analytics are going to be critical. And, brought it up, who's our audience? This is, an important handle. This and a lot of the presentations, even the artistic type of processes that are going on.
Well, that's really interesting. And this idea that you can't characterize, it's like the 95th percentile, right? So you have, the 95th percentile, nobody is that. Everything is around that.
Yeah. It
comes out on an average to that. How do we avoid this sort of misconception that people are sort of rubber stamped, you're human, so therefore you're like this, all of this kind of thing. So that's really an interesting comment. I guess that brings up another issue, which is the problem of how do we understand the things how do we help people understand the things we don't really understand, that there's complexity and there's uncertainty, and how do we communicate that this is not, it's not a simple thing, that these really difficult concepts that most people that people struggle with. How do we help them deal with that? Does anybody have a comment about that? Joe.
Science museums struggle with this constantly. People don't understand science as a living, changing thing. They understand it as a book, a set of facts. And I find this work mind-blowingly amazing and wonderful. And we keep continually uncover more complexity. People can understand things are complex if we help them understand they're complex, and that they don't need to know the answers. They just need to understand the complexity. For me, watching these amazing visualizations, I kept thinking, the body, functional parts of the body, from cells to organs, are in and of themselves beautiful. But then thinking about the surprise of, but it's beautiful in an art way, but it also is beautiful in how it functions and what it does. How do we help people make those connections, that getting drawn into this beauty is really opening the door to understanding the complexity of the human body, and we don't understand it fully, but isn't it a thing of art?
Yeah, that's a really interesting idea, and that things are not just by themselves beautiful, even if they are, but they're beautiful as part of something, and the way that they function. And I guess that brings up another sort of question about the value. So thinking about, we've actually all had this discussion at some time or another before, is how do we get people to understand that these things are not just valuable for you to know, but they're valuable to scientists and to artists and to humanists in ways that help them help us understand things. So it's not just trying to make a straight line between some concept and the public. It's also understanding that it's not just the public, it's about the professionals that have to deal with this. And they may be off doing something, but it's important for us to understand what they're doing and why they're doing it. That it's not that it's not that just important for us to understand something. I always talk about the Golden Fleece Award. It's William Proxmire used to do. I'm just showing my age. They give out the Golden Fleece Award to research that he thought was trivial. And one had to do with the ovaries of penguins being studied. And it turns out the ovaries of penguins are very similar to those of humans. And that was the reason for studying them. But people didn't understand that. And they thought it was laughable that someone was studying the ovaries of penguins in Antarctica and spending the public money on it. So it's important that we understand how important these things are to the scientific world and the public in general. Anyone else have a comment related to that? I know we're getting close to the end.
I thought it was really interesting hearing Weidi's perspective on the boundary between the body and the environment. I think that's maybe one example of a situation where art can maybe bring us a new conceptualization of how we're looking at the body. I mean, if you look at, human biome stuff, if you're looking at like the gut research that's happening, I think we are more a part of our environment than maybe we have thought of ourselves as in the past. And so anyway, I think that's interesting to look at those concepts that cross between art and science and all the other disciplines. There's that cross discussion is so fruitful.
Yeah, there was a comment in the chat about the problem of people understanding what scientists do and the uncertainty around science itself, not just that we're trying to communicate things that we don't know a whole lot about, but that science by its nature can be uncertain. And sometimes the things that we're absolutely certain of change over time, but trying to communicate that to the public by itself is a difficult concept, and that applies to all of science. So thinking about the complexity of communicating this, I mean, it's all sounding a little daunting, but I think if we think about how do we understand not just a particular kind of science, but the process of science itself. I think that's what the participant was talking about. So we have about 3 minutes left. Does anybody have any parting comments or things they want to add to what they've observed?
I'm curious because the term atlas has certain connotations in and of itself, which is that people look at the atlas and they think that it represents the body of knowledge. And it's continually changing, number one, and there are mistakes. and as you said, not all data is created equal. So, the term atlas might not be the best term for what is being done, even though at least, you can stake a name, but it should be more than that, actually. And it is more than that. From what I've learned today and seen, it's more than just a body of facts. In fact, what it should be is a repository, an interactive repository that people can add to and take away. I mean, just as an example, when people were starting to solve crystal structures, they developed an organization to archive those structures. It wasn't meant to distribute the structures. It was called the Protein Data Bank. Now, the Protein Data Bank now has 200,000 proteins in it, and now AI is using the Protein Data Bank to predict structure from sequence. So things change over time, and the functions of these organizations change over time. So I think we ought to be looking forward to how people can actually synthesize the data in new ways.
That's a really good point, Art. And I think that brings us to 9 o'clock or 10 o'clock where you are. I'm in Chicago right now. But thanks, everyone. This has been an amazing time. This has been recorded, so I guess we can go back and I think this has been terrific.
Thank
Thank you so very, very much, everybody. I learned a lot, and I hope we find ways to stay in connection. This was really, really wonderful. And I think also some of you might like to talk more. I see amazing installations coming up with combined forces that you presented here. Thank you again.